All right, well, thank you everyone for joining me today. It's quite a pleasure to be presenting some of my doctoral work. Some of this has been plaguing me for a few years since I've been starting it on my doctoral degree. So this is stuff that I did with my advisor and some colleagues, so it's exciting to kind of see this to fruition. I guess one more announcement is seepage is going on, so please sign up. It's this Saturday. If you're on the live stream, it's in Tennessee. All right. But what I'm interested in understanding is to apply phenotypic selection analyses to understand how we can generate morphological variation. And these are to answer some pretty fundamental questions in biology. So how did organisms come to look so different, and why do we see so many different types? Even some of the most oddest looking traits have persisted over time because they provide some kind of fitness advantage, like territoriality, or being able to move faster, either on land or in water. And these type of phenotypic traits can determine whether you die, like this poor hapless seal here, or if you go on to live another day, you potentially escape a predator. And these interactions can be quite complex between how morphology is generated. And this is a little tribute to Liz Hoban's awesome talk on social complexity in birds, but it's also to highlight that you can get some pretty weird phenotypic traits through selection. This is an example of sexual selection. But natural selection is a primary driver of adapted evolution. And here's a classic example where we're looking at the morphology of beaks in these Darwin's finches in the Galapagos. And so what we ended up having is that there was a single common ancestor, and eventually it led to an adaptive radiation of various species, and they differed in not only their beak morphology, but this beak morphology facilitated being able to eat different types of food, so different ecological niches that are being explored. And our understanding of evolutionary biology, including phenotypic selection, has benefited tremendously from evolutionary synthesis studies. And there's been various periods, so I'll just be talking about four of the main ones. First, we have this classic period, which was started by Darwin and Wallace around the mid-1800s, which really catapulted major concepts in evolution, such as natural selection. And then we had the population genetic synthesis. This is a relatively short period led by Jansky, Fisher, Wright, and Haldane. And then a common one that most of us know about is modern synthesis. So this is a pretty big one that occurred a few years afterwards, and this was led by a number of prominent evolutionary biologists and was to hopefully bridge a gap between Mendelian genetics as well as natural selection. And then recently, Steve Arnold proposed that we are currently undergoing another synthesis period. And what this particular period is pronounced is that we have even more data to be looking into these broad evolutionary patterns. We have a better conceptual framework in which we can start to tease apart different models to evaluate how evolutionary processes occur. And there's even better ways of testing these models using different computational methods. So mega data sets are really kind of at the forefront to help us understand these broad evolutionary patterns. And when we consider this major surge in the number of citations, so this is kind of a proxy for just how much progress has been occurring, we see that this major progress has been occurring due to a compilation or a combination of a lot of different fields. It's a very diverse synthesis. So it's including various aspects of biology, paleontology, genomics, but also mathematics. And phenotypic evolution is a primary area of research that Arnold is interested in. And one of the major milestones that occurred during the ongoing synthesis is this period of being able to use a theoretical framework proposed or developed by Landy and Arnold to evaluate microevolutionary patterns, so phenotypic selection, for instance. And we can look at those phenotypic changes within a generation. And these provide key parameters that can help in informing other types of analyses. So for instance, by using the Landy and Arnold method, we can start to get estimates of how strong selection is. And these estimates can then have downstream effects because they can be input as key parameters into understanding macroevolutionary processes. So the use of large data sets to test alternative models in evolution has provided a lot of insight into our current knowledge, and is helping us towards developing a much stronger understanding of phenotypic selection. So in today's talk, I'll be briefly discussing just some of the historical background behind how mathematics and statistics has played a role in strengthening our understanding of phenotypic selection. The lessons that we've learned can then be from applying those quantitative tools. And then I'm also going to be talking about some of the current challenges that we're facing. And so I'm just going to start with some of the earliest stages of quantifying phenotypic selection. So this is a relatively simple example. What we're looking at here is we're going to focus in on that Darwin's finch example. So here we have some birds, and we're going to see that the beak depth, so how tall the beak is changing. And on the x-axis, we have the depth of the beak in millimeters, and the y-axis is going to represent just the frequency. So on the top graph here, we see a relatively normal year. And we see that the mean trait value in the Z bar here, it's about 9.5. But then we had a selective event. A drought ended up happening, and that killed off a good majority of the birds. And what ended up happening is that when they looked at the mean value of the beak depth, they saw that this value shifted to around 10. So Z bar star is just at the mean value after selection. And so we can see that there's a deep statistical underpinning for looking at selection studies, because we're looking at changes in phenotypic trait distributions. So let's start off by looking at these hypothetical phenotypic distributions on this top here. So this is before selection. So similar format as we had before, trait's going to be on the x-axis and frequency is going to be on the y-axis there. And what I want you to look at is that there's different ways to generate more phenotypic variation. So for instance, when we look at directional selection, which is on the left of this dashed line here, it's also called linear selection because of that linear fitness function. We can see that this ends up changing the mean of that phenotypic trait distribution. But when we start looking at nonlinear selection, we start to see different changes to that trait. So what we end up starting off here is just normal traits, but then nonlinear selection can influence the variances. And then when you're looking at pairs of traits, you end up studying correlational selection, and what that ends up doing is changing the covariance between pairs of traits. And then in 1983, Landy and Arnold revolutionized the field by providing a theoretical framework in which that you could quantify the strength and the direction of multiple correlated traits. And so we'll start off by looking at an example for a two-trait system. It's fairly small, but it gets messy once you get more than two traits. So first we're going to look at linear selection. So directional selection, changing the means. And we have something called a selection differential. It's going to be represented by this little s here, and that represents the total amount of selection that's operating on a phenotype. And the nice thing about the Landy and Arnold method is because the computation is actually relatively simplistic. So here you can see that you can calculate the selection differential based off of your trait value, and then also your fitness relative to the mean of the population. Alternatively, you could also do something similar to what we were looking at with the finches, which is just to compare the mean traits before and after selection, so Z bar and Z bar star. Either way you do that, you end up with a vector of differentials. But the problem is that when you're looking at the output from the selection differential, sometimes it can get a little bit not confusing, but what ends up happening is that the selection differential has the effects of correlated traits. So when you're trying to understand the direct effects on an individual trait, the selection differential isn't really going to give that to you. What you end up needing is something called a selection gradient, and that's going to give you the direct effects on a phenotype based off of phenotypic selection. And for linear selection, it's going to be represented by this value beta. And you can calculate it either by using linear algebra and taking a phenotypic covariance matrix of your traits and then multiplying it by your selection differentials. you end up with a vector of gradients. But one of the really nice things is that you can also just calculate your beta values from an ordinary least squares regression. And this is a very common way to calculate your beta values, given that you're working with longitudinal data. And what this means is that you're looking at the change in traits within a group of animals over time. So this is only true for longitudinal data. All right. And you can use similar approaches to understand nonlinear selection. Here again, we have equations for selection differential, which is the total selection operating on a phenotype, but then you also have the selection gradient, which is the direct effects. What you'll see is that the variable did change, though. So for this selection differential for nonlinear selection, it's a C. But a very similar process is used. You end up using matrix algebra, and you can multiply it across using your phenotypic covariance matrix before and after selection. And this term right here is just to correct for directional selection. And so you see at the end, it's also based off of covariances between a relative fitness and trait value. So pretty similar to what we saw with linear selection. When you get down to the selection gradient, it's going to be represented by gamma for nonlinear selection. And you end up using either the phenotypic covariance matrix, as long as it's your selection differential, or you can estimate it from an ordinary least squares regression. And what you can see is that this is just a 2-trait system, so it looks relatively clean, but you can imagine how this can get really messy when you're starting to work with, say, 10 traits. This matrix is going to expand quite quickly, and so you run into an issue of a lot of parameters, and I'll talk about that more later. So the ease of this Landy and Arnold method has produced a pretty easy and quantitative method in which that we can analyze phenotypic selection. And it's resulted in hundreds, thousands of additional studies. And it really contributed a lot of information about how we're generating phenotypic variation. So in the next portion of my talk, I'll be describing what we've learned in the 30 years since Landy and Arnold first developed that method. And I'll be talking about a couple case studies that I worked on that have provided some insight, but is also some general information. So there's been numerous studies that have indicated that various biological factors can influence how selection operates on phenotype. So there's a lot of ways that you can generate diversity. The first example, Bankman recently did a review in which he found that the strength of a species interaction will influence how selection operates on phenotype. So in his review, what he found is that when you're looking at, say, a relatively, I guess, calm interaction, whether you're squabbling for food here, that's going to have a very different effect than if you had, say, a death match between 2 elephant seals here. And what they found is that as an antagonistic interaction, so kind of a negative interaction here, as that interaction becomes stronger, you end up having greater opportunity for selection. And so what this represents is that there's more chance for selective change to occur on that phenotype. And also what you end up having is stronger strength of selection. So there's a lot of ways in which you can generate more phenotypic variation. And sort of a very commonly studied system as well is to look at the effect of predators. So you can either just add a predator into a tank, or you could just even put its scent with the prey, and you can start to see that there are certain phenotypes that will emerge. So if you use a selection study, what ends up happening is that in the presence of predators, you might start to see that particular morphologies will be favored. So for instance, larger tails that will help you to swim faster, or larger bodies so you escape that size limit of the predator's mouth. And then a commonly studied area is also locomotion. Now, one of the reasons why locomotion is particularly important is because a lot of activities, a lot of daily behavioral activities depend on your ability to move around. So either escaping from predators, acquiring food, migrating, these all depend on your ability to move somewhere. And ultimately, your ability to move or to escape from predators, acquire resources, that's going to influence your reproduction and ultimately your fitness. So The morphology performance fitness paradigm, so this is just an axis here, what it essentially says is that there are particular morphologies that are associated with higher fitness. So that's a pretty general framework. But what they found is that behavior can mediate how this particular relationship works. And so what they found in the past is that behavior can mediate different forms of selection, and that can aid in evolutionary change. So a common example is the lizard ecomorphs. So on the right here, what we have is just a diagram illustrating various types of anolis lizards. You've seen them around here, the little green lizards running around. But in this system, what ended up happening is that there was some diversification because different groups started occupying different regions. So these are different species here. So some are better to move on the trunk, some are much better at moving in the canopy, and then others, kind of more standard, are better at moving in the grass and brush. And they're all associated with different morphological traits. So you might have longer legs here to run around, or you might have stouter limbs so they can cling onto the branch a little bit more. So not only behavioral differences, but also morphological differences as well. But these are relatively broad-scale differences in behavior. And so one of the things that we were interested in understanding is what about more fine-scale variations. So a previous study, they also looked at different anolis lizards, and they saw a differentiation of these lizards in different areas. But something weird that came up was that the aquatic anoles, you would assume that they would be pretty similar, but due to microhabitat differences, they were actually quite distinct in terms of their morphology. So some were a little bit closer to the shore. So the hypothesis was that just due to very slight differences in their locomotion, they were able to diversify. And so what we're interested in understanding is, if we're looking at finer scale differences in locomotor behaviors, how does that influence how selection operates on phenotype? And so to address that, my colleagues at Clemson University and St. Cloud University have used a system of kind of weird fishes. They like to climb waterfalls, or not like, but they do climb waterfalls as part of their life history. And the reason why this is of particular interest is because when you're looking at selection or trying to understand its effects, what ends up happening is that if you're looking at a longer time scale, there's more chances for different agents of selection to start operating. So if you're trying to hone in on a very specific aspect and trying to control it, it's recommended that you look at something that's fairly brief. And another feature about this system is that this is a pretty intense selective pressure, and I'll explain a little bit why. So to give you a general background about what the system is like, these animals have an amphibious lifestyle, so kind of like salmon, but different. So these guys, they end up living in fresh water. They lay their eggs, but then the eggs hatch, and then the water flushes the larvae downstream, and they move into the ocean, and that's where they feed and grow for a few to several months. Now, when they return to the islands, what ends up happening is that they're confronted by these predators. This is one of them here. Doesn't look that menacing, but trust me, they're pretty voracious. So they're onslaught by these predators that they have to evade. And in order to get away from these predators, they climb waterfalls. So this is an example from Hawaii. And the benefit of climbing these waterfalls is not only are they escaping these predators from the estuary, but they're also reaching these breeding habitats up above the waterfalls. And so I was trying to figure out a way to kind of illustrate the dire, I guess, situation that these animals face. And so I guess since Halloween's coming up, I guess I could say it's kind of like The Walking Dead. I know it's pretty bad, but just think, just wait. So what ends up happening is you get barraged by a bunch of predators that want to eat you. But what has Walking Dead taught us? Zombies don't climb. Neither do these predators. So this is kind of the situation that they're running into. You can try and live down here, but good luck. Your best bet is to get up above the waterfall in order to survive. But there's more than one way to climb a waterfall. And so we looked at two species from sister genera. There's Siceopterus simpsoni. It's called the incher, and that lives in Hawaii. And the reason why it's called an incher is because it moves like a little bit like an inchworm. But then we looked at Sycydium punctanum, which is our power burster here. and it lives in Dominica. And just to kind of illustrate what they look like in terms of climbing, what you're looking at is a view from underneath the fish, but it's climbing upwards. So look kind of at the side. This is some water here. This is a little grid. This is 1 centimeter. So to give you perspective, I think that's half. This little animal is only about 2 centimeters tall or long. And for the incher, what you're looking at is that it's using its mouth and a little suction disc in its stomach, or on its stomach. It's a pelvic suction disc. So it's climbing a waterfall with a mouth and a plunger stuck to its stomach. So this guy is kind of slow and steady. It doesn't really move its body that much. It doesn't use its fins. It's more similar to what a tortoise would do. Well, it's a slow and steady approach. But when you're looking at Cystidium punctanum from Dominica, it's a little bit more like the hare. It uses bursts of active activity, little sprints, and then it rests. And it uses a lot of movements of its body, so very different from what you're seeing here. It also uses its fins. I know it's translucent here, but its fins are out right now. And so it tends to sprint up, stop, sprint up, stop. So it's got more of a hare approach. You can see it wiggling a little bit there. So we have these, even though waterfall climbing seems to be a pretty distinct event, you can actually have very different ways to go about it. And what we're trying to figure out is, because it has this different behavior, how does that actually influence the targets of selection? And what we end up doing is looking at morphological traits that would be able to facilitate those types of movements. So here, what I have here is a lateral view of a fish, and then a ventral view, so looking from underneath the animal here. And what these traits relate to are either streamlining, so making it so that the fish can move through the water a little bit better, or using its sucker, that's what this little circular thing is, using that plunger to stick onto the waterfall a little bit better. So for linear selection, or directional selection, what we're expecting to see is that you would have larger suckers so that you can adhere to the substrate a little bit better. But also you want to be able to decrease drag. So having these lower values for your body measurements, so having a shallower body height or having a narrower body width, and then also being a little bit smaller, that can help reduce drag. And then for nonlinear selection, we saw this, the hare. It used a lot of coordinations between its body, its fins, and so the idea is that you're going to have more correlational selection operating on that system, because you do have more interactions going on between phenotypic traits. All right. So what we ended up conducting a artificial waterfall design. And so the reason why we didn't collect them out in the field is because there's a lot of variation in what you're experiencing out on these natural waterfalls. Especially in Hawaii, there's a lot of torrential downpours, so it causes a lot of variation. What we want to do is try and provide a very controlled system so we can do a direct comparison. And so what we ended up doing is having teams of about 20 individuals from each species climb up this artificial waterfall, which is about 100 body lengths in height. And to put this into perspective, imagine you have a little person down in red here, but they have to climb the Washington Monument. But not only do they have to do that, they have to climb a water slide up the Washington Monument. That's essentially what they're up against. So you have your life or death ultimately depends on whether you can climb a 500-foot waterfall. And the assumption of this model is then that reaching the top of this waterfall, which you can see here, that's going to give you higher fitness. Whereas if you stay at the bottom, that's going to give you zero fitness. That's where the predators are. Now this is just a proxy for fitness as well. So, and it results from the life history of these animals, where if they don't reach above the waterfalls, they're stuck with the predators, they're going to get eaten up. So after these experimental trials were conducted, we identified linear and nonlinear selection using logistic regression methods, and that allowed us to calculate selection gradients. So here on this graph here, we just have a phenotypic trait on the x-axis, and then relative fitness is on the y-axis here. The Landy and Arnold method typically uses just a linear regression, but as we can see here, when your fitness is binary, logistic regression curve tends to be a little bit better, and that's what Jansen and Stern did a few years back. And this will allow you to calculate individual selection gradients, but the problem is that when you're calculating individual selection gradients, you often underestimate the power of nonlinear selection. And so people have recommended that you calculate or you conduct a canonical rotation of that gamma matrix. So remember, gamma is that nonlinear selection gradient. So you end up with a matrix of it. Think about conducting a PCA. You're trying to get rid of the correlation between the traits. That's essentially what a canonical rotation is. And what this will do is then be able to identify your major axes of curvature. And what that means is that the output that you get from your canonical rotation will tell you, for instance, where peaks like this kind of occur. Will you find some kind of shape where it's domed like that? And being able to visualize the fitness surface has been a tremendous benefit in phenotypic selection analyses, because we can end up having a figure something like this. This is something that Chris Martin produced, and what he was able to do then is to produce a fitness surface and evaluate how these different types of species occupy different areas of that fitness surface, and even where there's a stable peak right there. And today I'll just be presenting just a subset of our some of our results. So overall, what we found is that these tortoises from Hawaii, these tortoise fish, they had a lot more individuals that were successful in climbing that waterfall, almost twice as much as the Dominican fish. And what these asterisks mean here are just that they were significantly different. And quadratic selection was not different amongst the two species, but directional and correlational were. So when you look across this chart here, you see that it's fairly similar in terms of what traits were under selection. Both of them had narrower bodies being favored, but this power bursting fish, also individuals that had a larger mass were tending to be successful, which is a little bit counterintuitive. Then we have correlational selection. What we find is that not only are there a greater number of traits under correlational selection for this power burster, but the strength of selection is a lot larger. So if you compare the average values, it's 1.5, but when you look at the maximum estimate of selection, it's almost twice. So stronger selection, nonlinear selection for this power burster. And that's what we also see when we look at these fitness surfaces. So again, we have the incher from Hawaii on the left and the power burster on the right. And think about these X and Y axes as kind of PCA or PC axes that are telling you about the multivariate traits of these animals. And the Z axis is going to tell you about your fitness proxy. So off the back, you can see that these fitness surfaces look pretty different. And when you look at the selection, I'm just giving you the strength of selection, we see that it's a lot stronger when we look at the power bursting animal. And just for comparison, this is about 0.4, that's about 4 times what we saw when we looked at only individual selection gradients. So already just by doing this canonical rotation, we're able to extract a lot more information about how selection was operating on phenotype. So we saw that differences in locomotor behaviors was associated with that were associated with a similar lifestyle did produce some variations in how selection operated, and we saw that the canonical rotation was actually more helpful in detecting selection, where the strength of selection was about 2 times stronger when we're looking not only at the individual selection gradients, but also even across the species, it was stronger in that power burster there. And correlational selection was stronger for that power burster, which provides support for our second hypothesis. And I'm going to talk a little bit about why you might expect that to occur. So what you end up seeing here is we've got the incher in green here, so that slow and steady tortoise, and then you have the power burster, so kind of that sprinting hare on the right here. And what I'm showing here is just the movement of different body parts. So we've got the mouth, the sucker, bending of the body, and the fins. And what you can see what you can see here is that the Hawaiian fish doesn't really use its body or its fins. It just kind of alternates between the mouth and the sucker. It's a pretty simplistic behavior. But when you're looking at this power burst, or here's just a reminder of the video, there's a lot of coordination going on. Before it moves, it's sticking with its sucker right now, and then it has to release its sucker in order to actually move forward, and then bend its body in order just to produce propulsion. But to prevent from sliding back, it has to time the attachment of its sucker quite carefully. And so you have a lot more coordinated movements along the body. And so there's a chance that the greater correlational selection is just due to the fact that you have more interaction going on in these different locomotor structures. So we saw that there is variation between species, but we also want to know how variation can occur even within a species. So if we're looking at, say, a single species in Hawaii, we'll stick with the slow and steady tortoise that we were talking about, Siceopterus. What happens if we look at different populations when they're under high gene flow? How do we maintain population differences? And so the Hawaiian archipelago has been known for its rich biodiversity and endemism, but the freshwater fish system isn't really part of that group. You end up only having about 5 native freshwater species, and it might have to do with the lifestyle that these guys have. So what ends up happening is that they flush their larvae out into the ocean, but then there's a potential for gene flow to occur. We actually don't know the water currents that well. But since they're mixing into this pelagic stage, there's a potential for a lot of mixing between populations to occur, because you don't necessarily go back to your natal stream. You don't necessarily always go home. And so, so when we looked at this fish previously, what we found is that the species on Hawaii, they tend to be a little bit more streamlined, but when you look at the same species on Kauai, they tend to be a lot deeper in their body height. So they look a lot more rugged, I guess you could say, on Kauai. So we're interested in figuring out, well, if there's such a high potential for gene flow in the system, why do we still see these population differences? And so local adaptation could be contributing to these to different selective regimes that could operate on their phenotype. Now, just to give you a little bit of a background, Hawaii is the youngest island with the archipelago. And what you end up seeing is that the waterfalls are really close to the shore. You can't even really see it on this map here. They're only about half a kilometer inland. And so this estuarine period where they're introduced to predators doesn't really last that long. They reach the waterfalls fairly quickly. So the primary selective agent is expected to be waterfall climbing, and that would select for more streamlined morphologies, because as you're moving through a water flow, you want to decrease drag. You just essentially want to look like a little torpedo. But when we look at Kauai, it's a lot older, and there's a lot more time for erosive processes to start influencing the geography or the topography of it. And what ends up happening is that the waterfalls are very far inland. They're about 30 times further inland than they are in Hawaii. So there's a lot more exposure during the individuals are exposed to a lot more predators during that time or for a longer period. So imagine essentially having to swim a marathon with these predators before we even reach that waterfall. And so in this case, we expect to see that there's going to be taller body morphologies, because tall bodies are good at producing thrust underwater. So by having this type of morphology, they're able to produce really fast bursts that they can get away from predators. And this is a very different contrast to what we would expect in Hawaii, the streamlined little torpedoes. So I'll be talking about a study where we looked at how individuals on Kauai exhibit different patterns of morphological selection. And what we expected is that there would be greater differences between islands than within an island, and there'd be greater opportunity for selection on Kauai because they're not, they may have a morphology that's further from a phenotypic expectation for waterfall climbing. So in Hawaii, the premium is for climbing waterfalls, but on Kauai, it's for predator evasion. So they might not be so good when they have to be introduced to a new selective agent. So we use similar selection analyses to look at linear and nonlinear selection, and we had 3 populations, we had Hakalau from Hawaii, which will be in green, and then Wailua in purple, and Waimea in blue from Kauai. And I'll be using this color scheme for the rest of the talk. But very similar apparatus here. So again, climbing up very, very tall waterfall for them. And then we looked at different morphological traits. These are slightly different from the ones that we used before, but they do relate to substrate adhesion as well as streamlining and thrust production. And we expect that the Hawaiian animals would have more streamlined bodies. Kauaian animals would have morphologies better for thrust production. And we use similar analyses that we used before, so looking at selection gradients based off of a logistic regression, and then also producing our fitness surface. But we were also interested in calculating the opportunity to selection, so to understand what's your standing variation, because that might be influencing your ability or the capacity for selection to operate. So think about opportunity selection as essentially the maximum amount of selection that can occur in a population. And I'm just going to be presenting just a few of the results that we've seen. So only quadratic selection differed on individual traits. Directional selection and correlational selection didn't differ, so I'm not going to be talking about that. And here we have the color codes again. And what we end up seeing is that these differences were mostly between Hakalau and green for sucker length, and then Wailua and purple. And the overall differences was primarily driven by these differences in the tail height here. So caudal peduncle height is also tail height. And the evidence for these population differences was also supported by these different morphologies or types of fitness surfaces. So Hawaii is going to be on the top, Wailua on the bottom left, and Waimea on the bottom right. And what I want you to do is start, I'm going to play a little video of it, and you're going to see how they look a little bit different. So as we rotate them, we can start to see that Waimea looks like a little bit of a cupped hand. This kind of looks like a swimmer doing a butterfly. Hawaii has a little bit of a sigmoid shape, and notice it's the only one that has a stable peak there. So I'm rotating them so we can get a bird's eye view and start to look at compare the topography of them. So this is just another representation of it. And what the X and Y axes are, just those canonical axes. Think again, just PC axes and multivariate traits. And these heat maps indicate how strong selection is. So the lighter the color is, the stronger the selection. When we plot the individuals on these surfaces, we see that most individuals don't really occupy the highest areas of fitness. And here we've got that stable peak again. It's kind of circled right there. And what we find is that there's a greater opportunity for selection when we're looking at these two Kauaian populations. So there's essentially more room for improvement, I guess you could say. It was about twice as much as what you found in Hawaii. And this might relate to the fact that we're also seeing a lot more failure going on in the Kauaian populations. So what these data start to indicate is that they might be a little bit further from the optimal, I guess, phenotype for waterfall climbing, which makes sense because that's not their the main selective agent on their island. So when we look at these individual plots, what we see is that this stable peak is characterized by generally more streamlined body shape, and then the higher fitness, which you can kind of see here, longer suckers, so bigger suckers to adhere to the substrate, and then a little bit shallow body heights. When we go to Wailua, which is a Kauaian population, you start to see that the highest fitness is associated with taller body heights and slightly smaller suckers. Kind of similar results for what we're seeing with Waimea, except now we're looking at taller tails and then larger suckers. So the larger suckers is different from what we saw in Wailua. So there are some similarities between these islands, but they're also fairly distinct. So why would you have taller body dimensions for these Kauaian populations? Because they're under selection for climbing. So one idea is that the sucker could potentially facilitate or counteract having to, so you have the taller body heights, but then you also have longer suckers to kind of help counteract the effects of those taller body heights. So correlational selection can help in that instance. But when we look at when we go back to the previous study that we saw, we have the incher from Hawaii and we have the power burster from Dominica, and we saw that they had different types of climbing behaviors. So again, incher on the left, power burster on the right. When we look at the quine population, that's a little fish right there. When it climbs up, it's still using its sucker and its mouth like the Hawaiian population, but it moves its body more. It wiggles its body a lot more than what you see in Hawaii. So it's kind of an intermediate between these two. So it actually might be undergoing a type of selective regime that's a little bit more similar to the power burster, and that's why it has taller body morphologies, so it can produce more propulsion. So contrary to our previous study, quadratic selection was the primary mode of selection, and that was different amongst these populations. And compared to Hawaii, the quine populations had lower average fitness, not as many were successful, the strength of quadratic selection was stronger, and there's also a greater opportunity for selection in the quine populations. So if we go back to our hypotheses, we kind of found some support for greater differences between the islands, but we also saw that there's differences even within an island. But we did find support for a greater opportunity of selection for the quine fish. And so these have just been a kind of small contributions, or just a few of the contributions to understanding phenotypic selection. There's been a ton of research, which I don't have time to go over, that indicate how biology, ecology, and various computational methods are influencing selection estimates. So for instance, we saw that by doing the canonical rotation, we could actually estimate stronger signals of selection. And so there's been a number of synthetic studies that have looked at phenotypic selection, and they have produced a lot of really good information about broad evolutionary patterns, but then also identifying different gaps in our knowledge that are helping us to identify where we need to go next, which is what I'll be talking about right now. And so during these analyses, I ran into a lot of challenges that I'm going to be pursuing for my postdoctoral research, and I'll be talking a little bit about what that will be involved. So a really fundamental question is just how strong is selection? And a number of people have been trying to address this. One of the classic studies is by Kingsolver and colleagues, and they used a pretty large data set, and what they found was that selection was generally weak. There's also very few studies that actually quantify nonlinear selection, which is it's a little bit troublesome because nonlinear, by doing so, you might be biasing your estimates by avoiding nonlinear selection or not incorporating it. And then also a lot of studies have low statistical power. But then Hare afterwards was able to identify that selection was generally pretty strong. And it all has to do with how you're standardizing your traits. So Kingsolver was standardizing the phenotypic traits based off of a variance. So that's going to be x-axis here. When Hare took the same data and then converted it so that it was standardized by mean, they were able to detect much stronger selection. So even just a very small effect is having a fairly large impact on how we're able to evaluate phenotypic evolution. And then so this one's a pretty big one here. What they found is that the theoretical framework states that the relative fitness is based off of different components of directional selection, quadratic, correlational. And what I want you to notice is the half terms here. But in reality, what happens is that statistical programs won't incorporate that half into their estimates. So what you get out of this, out of the output for quadratic selection is only half as strong as what it really is. And what Stinchcombe and colleagues found is that during a brief period, 75% of papers in evolution, one of the prominent papers that publishes this type of work, was forgetting to double their values. So no wonder selection looks kind of weak in that instance. So there's a lot of different ways in which the computational practices that we're using could potentially influence the big picture of what we're after. And our understanding of nonlinear selection just in general is pretty rudimentary at best. And one of the most recent syntheses by Kingsolver and colleagues have identified 4 major areas that would be good to evaluate. And I'm hoping to address the last 2. So looking at the effects of what types of traits you're putting into your model, what types of fitness measures or how they're affecting things. Also, what are you using in terms of preparing your selection estimates? And also, we really need to characterize more nonlinear components of selection. So previously I talked about canonical rotations and how that was helpful in my instance, but it's not always. This is actually a pretty big area of debate right now, is whether you should diagonalize or not. So diagonalize is also synonymous with canonical rotation. There's some arguments for and against. Some people say that canonical rotation, since they're multivariate axes, they're better at estimating adaptation. But the problem is that a lot of people are saying that reduces your ability to interpret the biological relevance of the data set. So sometimes it's helpful and sometimes it's not, but we don't really have a standard measure as to when we should be using this type of process. And so what I'll be doing is looking at what the effects are. So looking at different data sets from Dryad and seeing how the characteristics of those particular data sets are actually influencing what you get as output. And then there's a problem with the number of traits. So what we end up seeing here is just the x-axis is the number of traits, but since we have correlational selection, which is this last portion here, you quickly get tons of parameters as you're increasing your number of traits. So previously I told you I used about 6 traits in my analysis. That's not what I started with. I started with 17. So you imagine I'm working with a lot of traits and the analyses just would not run. But there's the effect of missing traits. If you're overlooking particular traits that are relevant for fitness, that actually biases your results. So you're kind of you're stuck in this dilemma of how do you know to choose, or how do you incorporate enough traits, but at the same time, don't put too many, because too many, not only will they maybe not even run your analyses, but that could actually cause problems of multi-collinearity, which is a big no-no in selection analyses. So it's just the fact that we're limited to a smaller number of traits really limits our effect to look at multivariate selection. And they've found that a lot of people only end up using 1 to 4 traits, which is really, really, really small. And so what I'm hoping to do is to even go down to the very basics of what is a morphological trait. I looked at linear measures, which is what this is showing here, so just looking at distances. It's based off of a Euclidean matrix. But if you're looking at shape, you don't really retain that geometry. What you end up having to do is something called geometric morphometrics, which is more of a Cartesian approach. And so it retains the overall shape of the animal. And a recent study by Collier and colleagues was able to evaluate the effects of or high dimensionality. So they have this fish, you put little landmarks to identify the shape, and they put a lot more landmarks on one of their data sets, so 10 versus 56. And by using this non-parametric approach that they developed, or the framework for it, they were able to provide new insight on the separation of sexual dimorphism in different habitats. So these new computational methods are allowing us to reach new heights in terms of looking at multivariate selection. But this was based off of just morphology. What about selection analyses? Considering that most studies have low statistical power, can we potentially use some of these other techniques that will be able to provide new insight and kind of overcome these issues of low statistical power? So what I'm hoping to do at NIMBioS is to essentially systematize the workflow for quantifying phenotypic selection. So there's the issue of just prepping your data. Are you transforming your data? Some people, to reduce dimensionality, they use PCAs and use those as traits. And then linear versus geometric morphometrics. Once you even get your traits, then there's a lot of areas that you can potentially process it. For instance, the mean versus variance standardization. And then there's a lot of options in terms of how you're running your selection analyses. Each one, so OLS is a landing arrow method, each one adds a different strength. But right now, what I was hoping to do is take a single data set and run it through this system and just start to evaluate how changing these various different parameters are going to influence your results. And the hope is that I'll be able to develop a graphical user interface, a GUI, in R that will really be easy for users to run all these analyses and really push forward in terms of using large data sets to look at broad evolutionary patterns. So that's the hope. And what I'm hoping to do is essentially facilitate those meta-analyses. So can we get better approximations of how strong selection is really operating, and what are the consequences of using these different types of methods? And so with that, I'd like to thank various people, including my collaborators and various colleagues who have helped me in terms of putting my ideas together. Also Brian Amir and Jim Fordyce for agreeing to be my co-mentors, so being very supportive of me, and thank you all for your time.